So the gene revolution is really a tale of two molecules. I'm going to talk about DNA, of course, that's genetic engineering at length. And when I'm done with you, you'll know how to spell DNA, I promise. The other molecule is a very small molecule. It's glycine methylphosphonate. It is a chelating agent. The name glycine methylphosphonate has been shortened to glyphosate. It is the active ingredient in the herbicide Roundup. So this is what I'm going to do tonight. I'm going to talk about what GMOs are, and then how they are created, and how they perform, what do they deliver. Because the industry tells us, the biotech industry tells us that they reduce the use of pesticide, that they increase yield, that they have no effect on the environment, and of course they are perfectly safe to eat. And I will review all these. And then I'm going to spend some time sharing with you a bit of recent research on Roundup. So what are GMOs? Well, over 90% of all the plants engineered on the planet today are engineered to resist the herbicide Roundup. Over 90%. So really, you could say GMOs are really about selling a chemical, Roundup. The technology was invented specifically for that 20 years ago. So we have Roundup Ready corn, we have Roundup Ready soybean, canola, sugar beet, potatoes, alfalfa, wheat, on and on. All the major crops have been engineered to tolerate the herbicide Roundup. There is another technology, another trait, which is really a minority trait. About 10 to 15% of the plants engineered also are engineered to become resistant to insects. They are called Bt. And Bt is the name of a bacteria, Bacillus thuringiensis. It was discovered 100 years ago in Japan by a German microbiologist, and that's why it's called Bacillus thuringiensis. Thuringia is a region of Germany. It was discovered at the time the silkworm industry in Japan was dying because there was some contamination, and these microbiologists determined what bacteria was causing it. They cleaned up their act and the silkworm industry survived, and everybody forgot about the bacteria until sometime after World War II, when somebody remembered this bacteria and it killed insects, particularly caterpillars, and wouldn't it be useful to use as a pesticide, particularly if it is a natural bacteria, which it is, and so we could use it as a biological control, as an organic pesticide, and that's exactly what happened. It was developed in the 50s and the 60s as an organic pesticide. The bacteria produces a protein which is incredibly toxic to insects. There is a whole family of bacteria producing a whole family of different proteins, and each one of them is quite specific. Some of them kill some caterpillars, others kill other caterpillars, and then the beetles, and then other insects. Some bacteria produce a protein, and we don't know yet what it is toxic to because we haven't discovered that yet. So HT stands for herbicide tolerance, and BT stands for resistance to insects. And these are really what genetic engineering is about in agriculture. Everything else is really, to me, a distraction. So when I hear about golden rice, and I know I'm in the Okanagan and I'm not supposed to say that, I'll say the apple, golden rice, the apple, drought resistance, and all kinds of other things in the future, feeding the planet, etc., etc. These are distractions. The reality today is about selling a chemical. Make the story really simple. And there is no doubt that the technology has taken over agriculture by storm. The farmers absolutely love it. They want it. To them, it's like, it's a wonderful technology. I mean, if you're a farmer and you're not using this technology, you are wasting money, you're wasting your time. The technology allows you, the plants have been engineered, when they're Roundup ready, they have been engineered to resist the herbicide, and so you can spray Roundup on them. and the plants stand up, but all the weeds disappear. Some of you are farmers, some of you are gardeners, you know what weed management, you know what the weeds can be. It's so much work. And as a farmer slash gardener, as you know, if somebody gave me a product that I could spray on my garden and all the weeds would disappear, I would love it. And that's what happened in farming. It makes weed management safe. The Roundup has next to no acute toxicity. When I was a graduate student, it was a new product. It was very safe. It was biodegradable. It had all the magic qualities. It was wonderful. And some of that knowledge has evolved a little bit over the time. I'll talk about it at the end of my talk. But there is no doubt that starting in 1996, when the first engineered crops were commercialized, they have gone up in 10 to 15 years, all the way up to 90 and 100%. Today, 100% of sugar beet are engineered, and over 90% of soybean and canola, and on, and cotton, etc. Which means that all these crops, by definition, are sprayed with Roundup, which also means that by definition, the food made from these crops contains Roundup residues. So that's what GMOs are. How are they created? There are two technologies to create genetically engineered crops. The first one is a natural bacteria. It's called Agrobacterium tumefaciens. It causes small tumors on the plant. And it does not infect all plants by far, just a family of plants, mostly the Rosaceae. So the apple, the pears, all the fruit trees, most of our fruit trees, the raspberries, the thimbleberries, etc., etc. And the way it does that, it does basically what bacteria do with each other. It does lateral gene transfer. Bacteria can transfer their genes to each other in a very simple manner. It's as if you were all bacteria in the room here, and I am a bacteria, and I have mutated. I now have a mutation of one of my genes, and I have a new protein. A protein that can do something that none of you can do. And I can do this really nifty thing, biochemically speaking, and you're all looking at me and say, can we have that too? And I said, of course, here, stand next to me, and I share my genes. That's what bacteria do, and this is what genetic engineering is about. It's called lateral gene transfer. And all the genetic information necessary for that gene transfer is on one of the tiny chromosomes that the bacteria has. Bacteria have a large chromosome and then tiny chromosomes, circular, which we call plasmid. And in this particular bacteria, there is a chromosome that has all the genetic information necessary for that transfer, and it's called the TI plasmid, the tumor-inducing plasmid. And when the bacteria comes next to the plant, it swims in the soil, and it comes next to the plant, and to transfer the genes, there are proteins on that plasmid that are activated and basically make a copy of a piece of the DNA of that plasmid. It's called the tDNA, the transfer DNA, and take that DNA and move it across the bacteria and into the plant cell and into the nucleus of the plant cell, and they cut and paste, basically, that's what they do, and they paste it and integrate it into a chromosome of the plant cell. And now that plant cell is engineered by the bacteria. This is the original genetic engineer. And that plant cell now must do what the bacteria says. And the bacteria says, feed me. And the plant cell is now making bacterial sugar that do not exist in the plant kingdom because of those genes, and gives the sugars to the bacteria. It's very elegant parasitism. And the next, there are only basically two ways. I mean, there are other ways, but these are the two major ones. The other technology is called a gene gun. It looks like a hairdryer, and you shoot lots of pellets, millions of little tiny pellets, almost microscopic, of gold or platinum or some metal that will not oxidize, on which you have coated the gene that you're interested in. And then you shoot. And one of the pellets, or possibly more than one of the pellets will end up in the nucleus of the plant cell, of a plant cell, and somehow by some mechanism that we don't know, and we certainly have no control over it, the gene will be integrated into one of the chromosomes of the plant cell. And now the plant cell is engineered with the gene that we want. So, okay, so now we have engineered at least one plant cell, but how are we going to find out which one? And the way to do that is to use a marker gene. And there are other technologies now to do that, but all the genetically engineered crops have been done this way. With the marker gene is an antibiotic resistance gene. Bacteria very commonly can become resistant to antibiotics. You all know that. It is easy to find the antibiotic resistance gene that allows them to be resistant, and you can take that antibiotic resistance gene and you can clip it out. This is cut and paste, and you put it next to the gene that you want to engineer, and you shoot the couple of genes into the plant cell. And so now the plant cell that is engineered is able to resist antibiotics. And what you can do is you can disintegrate the plant tissue that you shot to make sure that you only have the cells, not the cell walls, and you plate those cells on a petri dish where you have put some antibiotic. And with the antibiotic, all the plant cells will die except for the one that is antibiotic resistant. So now you have a plant cell that has the gene that you want. And from that plant cell, you treat it as if it was a stem cell or an embryonic cell with the right amount of different plant hormones, you can regenerate a whole plant. And then when you have a whole plant, you can multiply that plant, you can breed it with other plants to get other characters if you want, and then you can make hundreds of millions of acres of it. Today we have about 400 million acres of genetically engineered crops. This is how it's done. So what do they deliver? Well, the first promise, so to speak, from the biotech industry is that there is a reduction of pesticide. And it is true that for some years, until a few years ago, the BT technology, where the plant is its own pesticide, so to speak, because each cell of the plant makes the protein that kills the insects. And it is true that if the plant is its own pesticide, then you don't need to spray insecticide. And so there was a marked, notable reduction in the use of insecticides for the first 5 to 10 years of this technology. But not in the last few years, because the insects are becoming resistant. They have learned to become resistant to the technology. And the technology is failing. And there is nothing to replace that trait. And so now the farmers, where there is corn borer or other insects, the farmers are asked to spray insecticide, and the use of insecticide is now going up way more than we used to. With the herbicide technology, there has never been any reduction of herbicide use. Quite the opposite. The curves, of course, the technology has become extremely successful, so the sales of that particular chemical, have shot through the roof 1,500%, 15 times in the last 15 years. This is a really profitable market for the chemical company. And in the last few years, the sales are even going way higher, because now we also have weeds that are resistant to the herbicide. If you use, it's normal biology, if you use genetic pressure on a population of any organism, and you try to kill them, there will always be a few that are not quite killed, and they'll survive, and then next year they'll be even more resistant, and then they'll reproduce, and then you have a resistant population. And that's exactly what happened with the weeds. We have today over 40 species of weeds that are resistant to this technology all over the USA, and it's in Ontario, in Alberta, in Saskatchewan, in Manitoba. We don't have them in BC yet, and there's absolutely no reason to think that we won't have them this year or next year or next year. They're coming. Every scientist could have predicted that, and they did. 15 years ago, at the beginning of the technology. And of course, the company knew that that was coming very well. And so they've had several years to prepare. And so today, they are ready. Today, in the USA and in Canada, they are now marketing the next generation, the next technology. They have corn and soy, at least in Canada, corn and soy that are resistant to the next herbicide, and the next herbicide is 2,4-D. 2,4-D is a very old molecule, a very old herbicide. Many of you, I see a lot of gray hair, many of you are remember the Vietnam War, if you're not too old to remember. That was Agent Orange. 2,4-D is notorious for causing Parkinson's disease. So the farmers are going to pay the price. Everybody is going to have to pay the price. Not nice. No reduction in pesticide. GMOs increase yield. There is no way to imagine how this technology could increase yield. You are engineering the plants for herbicide resistance or insect resistance, and the yield is a very complex trait that involves hundreds of genes. It's like drought resistance. You don't engineer drought resistance or increased yield in a plant. This technology is good if you want to engineer a gene or two or even a handful, but not hundreds of them. This is impossible. There is no reason to imagine that they could increase yield except for good insect control from time to time in particular situations, or good weed control. But unfortunately, this is not what has happened at all, even with good weed control. This is don't bother trying to read the curves, it's too complex. This is the average corn yield in Europe. For all of Europe, average corn yield, this was compiled by a scientist in New Zealand and published 2 or 3 months ago. On average in Europe versus yield of corn on average in North America. These are published numbers, published statistics by the governments. And it so happened, and it's very obvious, it so happened that the yields of corn in Europe were lower than in North America 15, 20 years ago. And it's very obvious also that the yields in Europe are now higher than in North America 15 to 20 years ago. They do not use this technology in Europe. There is no increase in yield in corn. And there is no increase in yield in soybean either, and there's no reason to think that there's an increase in yield in the other engineered crops. This was a compilation published in 2009 by a scientist in the US showing data from the USDA, statistics from the USDA, as well as results of studies that were done at several universities in the USA, showing that basically the engineering process of the plant has a physiological cost. It is called a yield drag. And the physiological cost is about 5 to 10%. The plants that are engineered by definition yield less. And so the growers are basically trading for convenience of very good weed management and very easy weed management. The growers are willing to trade convenience, willing to trade a slight loss of yield. There is no increase in yield. GMOs do not impact the environment. I could approach this topic from several angles. I'm just going to do 2 tonight because I don't have a lot of time. And there's the first thing is called gene flow. And gene flow basically is when fields are engineered next to fields that are not engineered, be they conventional or organic. And of course the pollen flies around, whether with the wind or with the bees, and the genes from the engineered crops will fly in the landscape. And if you are an organic grower, growing corn perhaps, some of your plants will have been contaminated by engineered pollen. Some of your grain will be now engineered. And if you have bad contamination and you want to sell your plants abroad, or even locally as a organic product, if you have more than 1% contamination, you will lose your certification. And if you want to plant your grain and keep your seeds and plant your seeds next year, and if you are unlucky, Monsanto will come after you because you now have stolen their technology. And this has happened hundreds of times in the USA. And the organic growers are under a lot of pressure and basically terrorized by this company because of this problem. And we have in Canada at least one, there's probably many others, but at least one Canadian farmer who did not want to yield. He was not going to bow down, and he went all the way to the Supreme Court. This is Percy Schmeiser in Saskatchewan, who was growing canola and had some contamination on the side of his field. And he went all the way to the Supreme Court, and the Supreme Court said, this is Monsanto's technology, it should not be in your field. He lost a lot of money. We have lost our canola market to Europe and Asia, and we have lost our flax market. This is Canada. Flax market, also hundreds of millions of dollars. A lot of people today, because now we're just registering alfalfa in Canada, there's a lot of organic alfalfa, and a lot of people are extremely concerned because we're going to lose our markets, the same thing again. And I'm even reading that wheat has been engineered 15 years ago at least, but it has not been commercialized in Canada. And I just read something recently where apparently there's a push to commercialize engineered wheat, herbicide-tolerant wheat in Canada. And Canada is one of the biggest grain growers in the world, and we export 70% of our wheat, and we are going to lose our Asian market and our European market, and I think it is madness. This is gene flow. And gene flow can get even a little bit more twisted, and I call that genetic pollution. And genetic pollution is lateral gene transfer. This is what bacteria do to each other. The bacteria in the soil are perfectly capable of picking up the bacterial transgene. The genes in the plant, in the engineered plant, are bacterial genes. And those bacterial genes can find their way into the bacteria in the soil and then be distributed all over the field, the soil, the waterways, etc. We also have an experiment that was done years ago where people, volunteers, were fed engineered soybean, and the grains were not completely digested, perfectly digested in their intestine, and some of the DNA was still integral, and the bacterial transgenes were picked up by the bacteria in their guts. You all know that we have an immense number of bacteria in our intestine, so much so, and they are so important to our health, so much so that it is called an organ, it's called a microbiome. They are responsible for a lot of services to our health. For one thing, they're responsible for your happiness. They make 90% of the serotonin that goes into your brain. They're also responsible for making essential amino acid and vitamins, and definitely helpful for the digestion and many other processes. You want to keep your microbiome happy. This study was done in China. It was published a year ago, Sichuan University. We don't do this kind of study in North America. The Chinese were looking for antibiotic resistance bacteria in their rivers. And they found antibiotic resistant bacteria in every river they sampled. And the antibiotic resistance came from a synthetic gene, a blood gene. And that means that it either came from a lab or possibly, probably, from the local transgenic plants in the genetically engineered fields they grow a lot of engineered cotton in China. GMOs do impact the environment, And then finally, GMOs are safe to eat. Before 1996, when the first crops were commercialized, the biotech industry put forward a concept called substantial equivalence. And the concept of substantial equivalence works like this. Think of me as the chemical industry, the biotech industry, and today I go to the patent office, because everything is patented, believe me, and I go to the patent office, and when you go to the patent office to register your invention, you have to show, you have to prove that it is perfectly unique. And I go to the patent office and I say, I have this new variety of corn, and it is unique. There is no anything like it on Earth. And it has a new gene, and it has a new protein, and there is nothing like it. And I get a patent. And then the next day I go to the regulatory agency and I say, this is a new variety of corn that I've created, and it has a new gene and a new protein, and it looks like corn, and it tastes like corn, and it grows like corn, and it is perfectly equivalent. And in 1996, when the first engineered crops were registered, and until today, the regulatory agency says, Amen. Which means that the crops are substantially equivalent, and they do not require any kind of testing. No regulatory agencies in the USA or in Canada have done any kind of testing on those crops. And any testing is perfectly voluntary from the industry, and we don't need to know. And of course, it's top secret. When the technology was invented 30 years ago or so, it functioned with the old dogma, the old paradigm called the one gene, one protein hypothesis. It's really simple. That was the one gene, one protein hypothesis was put forward about 60 years ago, soon after we discovered how the DNA goes with the double helix. And it basically says that the genome is a linear thing with the genes, one after the other, and one gene codes for one protein. It's the one gene, one protein hypothesis. And in the 80s and 90s, some people started to see evidence that perhaps it was a little more complex than that. But really, it's only in 2002, the completion of the Human Genome Project, that we realized that actually this hypothesis has no reality. When we finished the Human Genome Project, we saw that a human being has about just over 20,000 genes. And most people agree, most scientists agree that we as human beings function with about 100,000 proteins. 100,000 proteins, 20,000 genes. How could that be? And what we realized, when I was a graduate student, the genome was, like I said, a collection of, the double helix with all the nucleotides, and you've got the genes, and then you've got all these sequences. They're long, long repeats, and they make no sense at all. And we don't know what they're about, and there's no meaning to them. It was called junk DNA. We have 5% of our genome coding for proteins, genes, and then 95% of our DNA is junk DNA. And in 2002, what we realized that for 20,000 genes to make 100,000 proteins, the junk DNA is very much involved. These are collections of regulatory sequences that tell the gene what to do. And the genes are playing with each other and collaborating. Today they make this protein, tomorrow this other one here, because the environment is different, and there's a lot of feedback, and it's a very, very sophisticated, complex, sensitive ecosystem. That's what the genome is. So you come with your gene gun and you shoot millions of pellets into the genome of a plant. And of course you're going to disrupt some of the sequences. But not only that, but if you forcibly inject the gene construct that you are placing the two genes that I took. There are other sequences with them. I'm making it really simple. You are going to put them somewhere in the genome at random, and they are bacterial genes, and they are completely different from what was there before. And Dr. David Suzuki describes it like this, and I won't roll my eyes like the way he does, but he goes like, the bacterial transgenes just go like, my God, where are we? What are we supposed to do? Because the regulatory sequences are completely different now. Their environment is very different. And it turns out that this process of genetically engineering a plant creates a lot of new proteins, not just the protein that you want, because the gene that you're inserting, you want that protein made. It could be the HT protein or it could be the BT protein, but a lot of other proteins are created. And many of them are, we don't know what they are. They are truncated, they're mutated, they are deformed, they're obviously, some of them will have some kind of enzymatic activity, and we don't know what they are. This is out of Belgium. Engineered corn, soybean, and canola all have variant protein. These are the new proteins that I'm talking about. Sometimes I call them rogue proteins, different from what the biotech industry reports. And these proteins are ignored by the regulatory agencies that said amen, and by the chemical biotech companies. 43 proteins in engineered corn plants were significantly disrupted compared to non-engineered plants. In bold here is normally the name of the scientific journal that the publication is in. In 1996, I already told you, this is when the first genetically engineered crops were commercialized. And what happened then, and I didn't know that, and nobody knew that until 2004, when this was published in Biotechnology, because two scientists at the time went back to interview all the people from the FDA and many other people and say, what happened? How come? And the expert toxicologist of the Food and Drug Administration in the USA spoke and said what happened at the time. And what they said was that they went to their director to warn him that there would be created many different proteins, truncated proteins, those new proteins, and some of them could or would be allergenic, toxic, create nutrient deficiencies and many other problems. And their director at the time was Mr. Michael Taylor, who had been a lawyer from Monsanto, and he had been put in place at the FDA to register those crops by the White House. And then when he moved along, sometime soon after that, he became vice president of Monsanto. It's called a revolving door policy. And he is back, he has been back at Monsanto for a few years just to make sure that the chemical company is well treated. And so what we have is this situation where basically the crops have not been tested in spite of the warning from the scientists, the toxicologists of the Food and Drug Administration. And then now we have a large body of science, quite a lot of studies coming out of outside of North America, because you cannot do this kind of research in North America because basically the company will not allow it. If you want to do research on those crops, you need to go to the company and say, I would like to do research, and you have to sign in blood that you will do the research according to what they want, and also you have to show them the data when you're done, and you will publish or not whether they, if they agree. So most of the research has been done outside, most or all of the research has been done outside North America, a great deal of it in Europe. And so now what we have is we have allergies, and we see the lab animal studies. They show allergy. This is the BT protein showing allergies, more of the BT immune response. BT corn causes anaphylactic shock.